Good evening, everybody. I'm Elisabetta Pupillo, and I live in Italy, in Milan. I work as epidemiologist in Italy in an institute. This is, as you can see, this is my institute in Milan. And here we perform a lot of pharmacological study, both in human and animal models. I'm very happy to be here today to speak with you and to explain the results of our research in the field of ELS, and in particular on the use of acetyl-Carnitine in ELS. So first of all, I will introduce to you something that is related to what we already done in the past. We will call acetyl-Carnitine as ALCAR. That is a acetyl ester of carnitine that increases intracellular levels of carnitine. And carnitine is a transporter of fatty acid across mitochondrial membranes. ALCAR, that is the same of ALCAR, counteracts motor neurons death induced by toxic agents or degradation of trophic factors, and it's able to slow the disease progression in animal models with a degenerative disorders mimicking ELS. We say that mimicking ELS because they are not a real animal with ELS, but we are able to use this kind of animals that are able to mimic the same symptoms of ELS with a specific genetic modification. The postulated mechanism of action of acetyl-Carnitine is complementary to that of riluzole, which suggests the possibility of a combined action. So we think that the use of acetyl-Carnitine plus riluzole can have an increased effect, beneficial effect than riluzole or carnitine alone. An additive effect of acetyl-Carnitine and riluzole was documented in the survival of transgenic mutant SOD1 mice. And this is the SOD1 mice in a specific model of mice with ELS, when the combination was compared to acetyl and riluzole singly, as already I explained. So what was our hypothesis? We hypothesized that ALCAR treatment can improve motor neurons activity, acting as a neurotrophic factor at mitochondrial and NMDA receptor level. So what we done already? We performed a double-blind placebo-controlled phase 2 trial. This was a pilot study. Very small study. We published the results of the study in 2013. It was a multi-center, randomized, placebo-controlled parallel arm. So that means that we involved 16 experimental Italian centers across Italy, and the subjects received carnitine or placebo at random. We included male and females aged to 40 to 70 years with defined probable ELS according to the revised editorial diagnostic criteria. We also included both bulbar and spinal onset, and the disease duration should be from 6 to 24 months. The satisfactory bulbar and spinal function were detected with the ELS FRS scale, that is a functional rating scale that maybe you perform at each visit at your site. And we requested that the item related to swallowing, cutting food and handling utensils and walking should be scored 3 or 4, not less than. The subject should have a satisfactory respiratory function. So we enrolled only those subjects that had an evaluation of FVC more than 80% as predicted. And also the subject should have documented progression of symptoms in the last 3 months. Also, of course, they must be able to understand and to comply with the study requirements, including informed consent signature. All the enrolled subjects received 3 grams daily of acetyl-carnitine or placebo in addition to Riluzole, and Riluzole was taken as 50 milligram twice a day. We performed a visit at the baseline, so day 0, then after 4, 12, 24, 36, and 48 weeks. And at each visit, we performed a general assessment, including the functional disability, so the ELF-FRS scale, the muscle strength using the Medical Research Council scale, that is scale validated to evaluate the strength of muscle, the respiratory function using a spirometer to measure the FVC, and also the quality of life using the McGill quality of life question. I know the question that today we will not more use this. We enrolled subjects between March 2007 and February 2010. The aim of our project was to evaluate which is the proportion of patients no longer self-sufficient after 12 months, defined as individual that reached the score of 2 or lower at the item of swallowing, cutting food, and handling utensils or walking related to the functional register. Secondary evaluation, so the evaluation had an important aspect, but was not our primary goal, were also the evaluation of the muscle strength and the ELF-FRS total score, the FVC score, and the proportion of cases desired or needed tracheostomy at the end of the study, and the proportion of therapeutic failures, and then the score, quality of life evaluation at the end of the study. So what is important from this slide? We enrolled 42 subjects that were treated and received acetyl-carnitine, and 40 subjects that received placebo. And the 2 groups were similar as at the first evaluation. So we performed a statistical evaluation to verify that both groups were similar, and the only difference was only the treatment that they received. And this was the case. So we can say that the randomization worked very well, because no differences were detected between groups. Nor in the demographic aspects, nor in the disease characteristics, and nor related to the evaluation performed at baseline. Then we performed the evaluation of the functional rating scales. We can see in these figures the 2 curves. The blue one are those subjects treated with acetyl-carnitine, and the red are those treated with placebo. And as you can see, during the treatment period, the people were treated for 48 weeks every day. We detected a trend, of course, because unfortunately we know that this progression, but the progression was lower in those treated with acetyl-carnitine than in those treated with placebo. And we detected more than 6 months difference in the functional decline between the two groups, with a significant correlation with time and time-treatment interaction. This means that the difference that we detected was due to time and due to treatment. So we can say that maybe the drug can have a positive effect slowing the disease progression. Also, we performed a lot of other analyses, also related to the spirometry, and we found that the forced vital capacity was higher in those subjects treated with acetyl-carnitine than in those treated with placebo. But we didn't find an interaction between treatment and time. So this means that the carnitine maybe has an effect also in the FVC, but it was not time-related. And from these studies, we can't say that carnitine has a real effect on lowering the FVC the evaluation. Also, we found that there is a trend in survival. So acetyl-carnitine, those subjects treated with acetyl-carnitine died less than those treated with placebo. And this was performed in all subjects involved. When we excluded those subjects that had a lot of protocol deviation, the subject that was not compliant with the treatment, of those subjects that dropped the study, so decided to interrupt the treatment, this difference was not statistical significant. So starting from these results, we decided to evaluate what happened in the general population treated with acetyl-carnitine, because when we published the results of this clinical trial in 2013, then in Italy, the neurologists started to prescribe acetyl-carnitine to the subject. But we were not sure about what really happened, which was the real effect. And some neurologists prescribed the subject the treatment with 1 gram, 1.5 grams per day, or 2 grams or 3 grams. So we decided to evaluate what happened in the real world. So what happened in the use of acetyl-carnitine as per clinical prescribed as per clinical practice. So 2 years ago, we published the new results from an observational retrospective case control study. So we decided to add more evidence and to perform additional evaluation on the use of acetyl-carnitine. We enrolled subjects, we checked the clinical charts of the medical charts of the subject in Italy, but we included those subjects that had 18 more years with a defined probably laboratory supported ELS. Again, we enrolled only those subjects that were self-sufficient, so with a score of 3 or 4 on the ELS FRS scale related to the item of swallowing, cutting food, and handling utensils and working. With the forced vital capacity that was more than 80%. We decided to use these inclusion criteria because they were the same used in the previous clinical trial. We divided the subjects, those treated with acetyl-carnitine at 1.5 gram or 3 grams or placebo. Sorry, not placebo, not treated with carnitine. We matched the subjects with the subject on treatment with acetyl-carnitine with those not treated, and the match was performed by sex, age, site of onset, and disease duration, in order to be sure that the baseline characteristics were similar. The objective of our study was the evaluation of the effect of acetyl-carnitine treatment on survival. The secondary objectives were the evaluation during the 12 months after baseline of the effect of acetyl-carnitine treatment on functional decline, on forced vital capacity, and on self-sufficiency. For patients with sufficient follow-up time, we performed also the same evaluation after 18 and 24 months after baseline. Baseline was the visit, was considered the visit where the subject started to take acetyl-carnitine. We included 180 people with ELS, and the characteristics of subject in treatment with 1.5 or 3 grams per day of acetyl-carnitine were similar in both groups. Again, I will not show here, but also we verified that these subjects were also similar to those not treated with carnitine. And so we can be sure that, again, in case of we detect a difference at the end of our study, our results will not be biased by general characteristics, but maybe the differences would be only due to the treatment. First of all, we evaluated the cumulative survival probability. So we evaluated that subject on treatment with acetyl-carnitine, so those in red, and subject not treated with carnitine, so it was in blue. The question was, are those subjects in treatment with acetyl-carnitine, has subject in treatment with acetyl-carnitine a better survival than was not treated? And when we evaluated all subjects in treatment, both 1.5 or 3 grams versus those not treated, we didn't detect a difference. So if you have you can say a difference there, because starting from month 12 we are able to see a different trend, and this is true, but the statistical evaluation say that this difference could be due to chance. Then we decided to perform other additional evaluation on survival, and we divided the subjects. We performed an analysis dividing those subjects treated with acetyl-carnitine 3 grams daily versus no treatment, and this is the scheme. Then we evaluated those treated with 1.5 grams of carnitine versus those not treated, and is the line in blue. And as you can see, we detect a difference. And in this case, the difference was statistically significant. This means that the treatment at 1.5 grams has an effect on survival. We don't know why the treatment with 3 grams in this case didn't show an effect. Then we performed an evaluation also related to the other parameters that I explained to you before. If you take the picture B, this was related to the self-sufficiency. So those subjects not treated with carnitine and those treated with 3 grams had no difference in the functional progression, and the same was for those treated with 1.5 grams versus placebo. So we didn't detect an effect on self-sufficiency, so on the autonomy of the subject. And maybe you can see it much better here, because this is the total score of ELS-FRS scale, subject treated with 3 grams and placebo, so the line are overlapped. So no differences in the total score was detected in those treated with 3 grams of acetyl-carnitine, while in those treated with 1.5 we detect an effect. Also, but this was not statistically significant. So related to the FVC, so the spirometry, we did not obtain significant results, both in 3 grams and 1.5 grams per day. Even if the slopes were virtually the same. So the general conclusions from these 2 studies is that acetyl-carnitine showed positive effect on survival, but these data are not in line with the previous findings. And we really don't know why, but maybe this could be probably explained by the retrospective design of the present study. So when we perform a retrospective design, we have not a regular collection of ELS-FRS score of FVC at fixed time points, because every subject performed the clinical evaluation in different time points. Sometimes we had information from visits a delay of visit from 1 or 3 months, subject to subject. So it wasn't nothing was very standardized. But the results of an observational study are very important because they add something that should be better studied. And so this is why we thought that a new randomized phase 3 clinical trial is needed, because we have to better understand the effect of acetyl-carnitine, both 1.5 and 3 grams per day. So we decided to plan this new study, and we asked for money from an association in order to be able to perform a new big study. And the primary aim of our study was to assess the efficacy of carnitine in 2 different dosages, so 1.5 and 3 grams daily, on the progression of functional disability measured by the ELS-FRS scale. We need a big study in order to test 2 different dosages. So we evaluated which is the best number of subjects to include in the study in order to have final results that can say as if carnitine is able to slow the disease progression and which is the best dosage to use. Also, we will perform additional evaluation to test the effect of ALCAR on other clinical aspects. So not only the self-sufficiency, but also the total score of the ELS-FRS scale, the decline of forced vital capacity, the quality of life of subjects included in the study, and now we will use the so-called ALSA-40 scale. So there are 40 questions that evaluate the quality of life of the subject, and also the cognitive function of the enrolled subject, and we will use the ECAS scale and the survival. But also, we would like to test also biomarkers to be able to evaluate not only clinical aspects, but also what happened inside the subject, biologically speaking. And we will perform evaluation of a list of biomarkers. Of course, we will also evaluate the tolerability and the safety of the drug. So we will evaluate which are, if any, the adverse events. So we will include 246 subjects in Australia, in 1 site in Australia, and more than 10 sites in Italy. And we will include subjects with similar characteristics with those enrolled in the previous study. Subjects must have the diagnosis of ELS according to the Gold Coast criteria, that is similar but different of the editorial evaluation. The disease duration should be less than 24 months. Again, we would like to enroll subjects that are self-sufficient, because we would like to evaluate if the subject treated in the moment in which they are able to be autonomous is useful to stop the disease progression. And this is very important because this is a disease we don't know very well the mechanism of action of the disease, and we don't know which is which are the cause of the disease and so on, I think that you know. And at this point, it's very important to start the treatment at the earliest moment as possible and in a good condition. And this is also why we would like to enroll subjects with a high level of FVC, so more than 80% of the predicted. And the subject must be in treatment with riluzole, and the treatment with riluzole must start at least 4 weeks before the inclusion in the study. As I anticipated to you, the subject will be treated with acetyl-carnitine 1.5 or 3 grams a day or placebo in addition to riluzole 15 milligrams twice a day. We will randomize the subject, so we the computer will decide which is the treatment to be assigned to the subject. And we will treat the subject, the enrolled subject, for 48 weeks. And we will follow the participant until death. We will perform a clinical evaluation at baseline, then at week 4, 12, 24, 36, and 48. We will perform a general assessment, a neurological examination, the ELS-FRS scale. We will perform the spirometry, the ECAS scale, that is the scale that evaluates the cognitive aspects, the quality of life, the compliance with the study treatment, and of course we will collect the blood samples to test biomarkers. As you can imagine, the number of subjects that we would like to enroll is very high. And based to this, we hope to be able to enroll a so high number of subjects, 246. But of course, we are not sure because this is a rare disease. So we decided to use a statistical evaluation that is called adaptation, study adaptation, and the specific name is pick the winner. So in case of the mean recruitment rate will be less than 4 patients per month, we will perform an interim analysis when at least 20 participants in each of the 3 treatment arms we will reach half of the treatment period, so 24 weeks, in order to evaluate which is the best treatment dosage that could have the best effect. This is a very complicated statistical evaluation, but using this methodology, if it will be needed, we can stop to use one dosage, and we will going on with those dosage that is maybe more promising, and with the placebo. Of course, those subjects that will be already randomized to the treatment dosage that we will not use more, those subjects will reach the end of their treatment period with the dosage with the original dosage. The subject enrollment will start in September this year, and we planned to perform the final statistical analysis, so be able to give the final results in June 2027. And thank you for your attention. I hope that everything was clear, but I'm here for you to your questions.
Okay, excellent. Excellent, Dr. Pupillo. Thank you so much for that presentation. Well, a really exciting study. So we hope it can bring positive results very soon. I just have 2 or 3 questions for you here. Well, first of all, we've heard about amino acids in the past. As a matter of fact, there is a very well-known website that usually analyzes different alternative treatments around the world, which is ALS Untangled. You must be familiar with it. And there is a report about acetyl-carnitine in ALS Untangled, and it says that it does show promising, it does show promise and potential for the treatment of ALS. But of course, as you also said, we need to have more clinical trials performed. We need more clinical trials in order to finally determine whether this has a benefit for our patients or not. So what can you say about the use of amino acids or a kind of amino acids like this one, like acetyl-carnitine, which is also available in the pharmacy? What would be your recommendation for patients that are, in the meantime, until we get results from your clinical trial, those patients that are willing to explore or to experiment with an over-the-counter purchase of these kind of amino acids?
So first of all, it's very important to have a conservative approach, because that's a lot of amino acids that were tested in the past in ELS, and there's also publications about, and we found that unfortunately those called ramificate amino acids where they had a toxic effect. So subjects treated with this kind of amino acids dead more than those in placebo. So we must use amino acid in a very careful way. Related to acetyl-carnitine specifically, I know that there were no serious adverse events related to the use of acetyl-carnitine. The most representative adverse event was diarrhea, and at the dosage of 3 grams. So maybe it is very important to ask the opinion of the neurologist in order to evaluate if in this specific case the use of carnitine would be recommended or not, because the ELS is a very heterogeneous disease, and unfortunately by now we are not able to say yes, this is useful for all the ELS patients. So what is very important is to ask the opinion of the neurologist or the general practitioner. And I can say that the tested dosage in clinical trial was 3 grams. maybe also 1.5 grams can have a positive effect, but we don't know. So in case a subject would like to start the treatment with acetyl-carnitine, can choose between 1.5 to 3 grams without any sort of toxicity or serious adverse events. But first of all, ask the opinion of your neurologist.
Okay, that was very clear. Thank you for that. It was important to talk about this topic. And so after the unfortunate failure of AMX 0035, and apparently we're not having very good results about oral edaravone. Unfortunately, the only drug for ALS that has strong evidence and it has been it's being administered for ALS patients for a long time is Riluzole. And in your clinical trial, as I could see from your presentation, you're really making a point about the combination between Riluzole and acetyl-carnitine. Can you expand a little bit more on that?
Yes, we detected in animal studies that there's a synergic effect when we use acetyl-carnitine in addition to Riluzole. And so this is why we decided to use a combination of both. For those subjects that can't use Riluzole, the use of acetyl-carnitine alone is recommended, but maybe the effect could be less than the synergic effect on the use of both the treatments.
Excellent, thank you. And so what are the next steps for this clinical trial? I mean, when are you expecting to publish next results? And when can we expect to have a potential maybe conditional approval by the EMA, by the European Medicines Agency? I know I'm trying to go a little bit too much ahead on time, but what can we tell us about that?
EMA request additional information, and so this is why we decided to perform our new clinical trial. So until the end of the study, we will not have any kind of approbation. But what is very important is that the company, so is owner of the company to request the approval to EMA. And we hope that the company will be interested in this. I don't know, because this is an independent study. So, of course, we are speaking with the company. They will provide us the drug and the placebo and so on. So we have a very good interaction with them. But then the final decision is from the company. Of course, in case of this trial, we say to us will show us important observation and results. I'm sure, so we will publish the results in case, and in case of positive effect, a confirmed positive effect, I think that EMA, first of all, will evaluate the possibility to prescribe acetyl-carnitine, not off-label, but as per clinical practice in ELS. In Italy, there's some administrative area where they already approved the use of acetyl-carnitine in ELS, but this is not law. This is a general consideration, but it's not a final decision from the Italian agency.
Okay, thanks again. One last question. I was going to ask you, how did you start to study, to become involved in the field of ALS, and how do you see the current ALS research landscape in the world?
So I started during my thesis period, just for, I don't know, for mistake, because I looked at the news in TV, and I was impressed by the relationship between ALS and soccer football players, and I thought, okay, I would like to better understand this, and so I started to study ALS during my thesis period, and I never stopped. I think that in the last 5-10 years, the collaboration between associations around the world and the researchers around the world is improved, and this helped us to perform more useful study with a large sample size, and with so that the interaction between researchers increased during the time, and this is a very good practice, and I hope that this will improve during the next months in order to speed the knowledge of this disease and to be able to find something very useful to all the ELS community.
Okay. So thank you very much, Dr. Pupillo, for your presentation and for answering our questions, for sharing some time from your very busy schedule, I'm sure. So we're looking forward to hearing more news about this exciting clinical trial. So thank you again. And if you want to give your salutations for our community to wrap up.
Yes, please. I would like to thank you, all of you, and I'm available in case of additional questions also next days in the future. And thank you for spending your time with me today, and thank you for your invitation. And so we will do our best for you. I would like to underline this because we really are doing our best, even if with a very few news, but it's very difficult, but we would like really to improve our knowledge to help everybody. So thank you for your invitation.
Okay, thank you, Dr. Pupillo, and keep up with the good work. Thank you for keep investigating for our patients, our ALS patients in Argentina, in Europe, and all around the world. We're looking forward to seeing you soon, probably in some scientific meeting. We have Montreal at the end of this year, so maybe we'll be there. Okay. Thank you, Dr. Pupillo, and take care. See you next time.
Thank you. Goodbye.
Bye. Thank you, Elisabetta. Thank you so much.
Thank you to you. Bye-bye.
Bye.